Hi, everybody. Thanks for your interest in my talk and in the educational course in general. So I'm going to be talking to you about tractometry and how we've used that to understand white matter development and some links to behavior. I also want to kind of make sure I'm talking about current concerns in the field or what's coming next, to kind of give you just a primer on the state of the literature and some tips on how to design future studies. Okay, so let's get into it. I want to start with a note about scanning kids. So I work at the Alberta Children's Hospital out of the University of Calgary. We scan a lot of little ones there, as young as 3 months of age and all the way up into the 20s, depending on the study. Scanning younger kids, it presents some unique challenges. As you can imagine, a 3-month-old or a 2-year-old or something might be a little bit less willing to sit still for an hour or a half hour at a time. But it is possible to get them in the scanner and have them have a positive experience with coaching. We've made our MRI look like a rocket ship and all that stuff. So we might be dealing with a little bit more motion or maybe a little bit less availability of data, but it's possible to get good data from young kids. And in fact, it's very important as a lot of the magic of brain development happens early, right? Okay, so with that kind of extra consideration, since we're talking about brain development, let's talk about tractometry. So we've found our tracts, we've fitted our diffusion models, we're confident that we're ready to do some analysis. What do we do next? There's a lot of answers to that question. That's probably not a surprise. I'm going to be focusing a lot on whole tract analyses. I think it's a good balance for this presentation to give kind of a broad overview. But it's worth noting that this one process of taking the corticospinal tract and turning it into a voxel-based mask, and looking at the average structure of that entire tract at once. That's a pretty broad approach, and it's only one approach. There's others that are more granular, like analysis along the tract, or tract-based spatial statistics, or voxel-based methods. Lots of other tools out there that are going to kind of augment the findings that I'm talking about today. So generally, analysis of tracts and developmental trends has been going on for quite a while. I've got this study from 2008 on the screen, showing that some tracts are finished developing by 13 years, others keep developing up into the 20s, and others are developing even beyond that. They're still going strong. The general trend in development, though, is fractional anisotropy, or FA, goes up, and mean diffusivity, or MD, goes down. These are the main metrics from DTI, diffusion tensor imaging, from that model that get discussed and published a lot, because they're nice, sensitive and broad metrics of white matter structure. Higher FA people generally interpret as more presentation of a more mature tract, or a tract that's further along its developmental trajectory. Same with lowered MD, but there's plenty of causes that could be behind that, which we'll touch on a little bit later. So there is regional variation in this trend, like I mentioned, and on the right, we can see some longitudinal data with males in blue and females in red, showing that sex differences in development are typically minor. There's individual differences in developmental trends, but it's typically not delineated by sex in any meaningful way. Sometimes sex findings may be reported, depending on the study and analysis choices and the regions being investigated, along with age range. Lots of different factors in play here. This kind of ties into my next point, which is that how we analyze our data does matter. So on the left, these are just some global trends in FA and MD and white matter volume for good measure. On the right, we have FA versus age in 4 different regions, with each colored line showing findings reported by a different study. You can see there's a decent amount of consistency in terms of the trajectory of development, but there's quite a bit of spread in the actual reported FA values between studies, depending on age range and analysis type. This is really interesting discussion of this in the source citation. One little thing I can pull away to mention here is that methods that focus on querying or investigating just like the central portion of a tract, the region that's most likely to be part of the tract. They tend to report higher FA values or lower MD values than whole tract approaches like what I'm focusing on, because the whole tract focus also includes voxels at the outside of the tract that might be partial volume effects may be present there with CSF or gray matter or what have you, and generally you see lower values reported for whole tract approaches. So analysis choices matter, and so does the age range we're looking at, right? If you're, say, for example, we have this global trend in mean diffusivity here from 2 to 8. It almost looks like there's, a trough, a plateau reached at age 7 for global mean diffusivity, but that might just be because our sample ends at 8 in this study. And if we continued further, that kind of uptick in our population level curve might not exist. So another thing to consider in terms of interpretation of our findings here. Okay, so links to behavior. This is a big topic. There's lots of different kinds of behaviors out in the world, of course. So I'm just going to give a couple examples and hopefully give you an idea of the general trend. So DTI metrics, FA and MD mostly, have been associated with a wide range of cognitive and behavioral measures. The general trend is, higher FA and lower MD. That's the trend we see in development. Those are linked to, presentations of more mature behavior. So better reading skill or, improved emotional control, things like that. So I've got some examples on the screen here. The first one at the top is showing different relationships between FA and age on a cross-sectional study in the ensign fasciculus for kids with higher externalizing or lower externalizing behavioral scores. If you're not familiar, externalizing behaviors are problematic behaviors that are, from the self directed out into the world. So things like aggression or defiance or, social opposition, things like that. And all of the kids on this, or all the volunteers in the study were subclinical. None of them are diagnosed with behavioral problems, but this relationship is still present in healthy developing kids, with a flatter relationship between FA and age, implying maybe a plateau in development, but this is cross-sectional, so we can't fully conclude that for kids with higher externalizing behavioral scores, whereas we see a more positive trend in those with lower behavioral scores. On the bottom, we have a longitudinal analysis of white matter structure and reading. So this was a study of kids entering adolescence. There's a 2-year period between their scans, and these scatter plots are showing that across that 2-year period, a greater rate of FA development coincided with a greater rate of improvements in reading scores, both in the archaeofasciculus and the inferior longitudinal fasciculus. Now, another interesting thing to note here is that the tracts that you are investigating and trying to kind of investigate links to behavior, the tracts you choose matter. So there's this nice quote from a 2021 paper mentioning that links between FA and behavior are particularly consistent in the corpus callosum and the cingulum, whereas other tracts might be, less consistent in terms of their reported associations to cognition or behavior. This is kind of a segue for me into talking about reproducibility of findings, which is a big focus in the field right now. Let me just get myself out of the way a little bit. So this is a big focus of, the field of brain and behavior relationships. There's some pretty high profile publications coming out on this topic, and it's a big focus of conferences like OHBM as well. But I will note that here, a lot of the focus of these publications, for example, is on things like gray matter metrics or functional MRI studies, and the considerations for reproducibility of brain and behavior findings still apply to tractometry. But in fact, there's this publication from Aperture down at the bottom that's specifically looking at brain and behavior links in white matter and finding that concerns maybe need not be quite as large as they are for functional or gray matter based approaches, just based on the way that analysis plays out in tractometry. But still the same best practices apply of, limiting the amount of comparisons or models that you're running, using kind of gated analyses to look at global or whole tract trends maybe before digging into a more granular or finer approaches, and leveraging longitudinal data or larger public data sets to kind of just include more data in your studies, and kind of increase confidence and statistical power, for your findings to be better supported. So reproducibility crisis, still relevant for tractometry and white matter analyses, and something that needs to be considered in terms of future study designs. Okay, so we've been talking about healthy development and healthy links to behavior in healthy kids. I also want to talk about atypical development. So I'm going to use perinatal stroke as an example. This is an image from a patient with perinatal stroke. Everything in white is a lesion. So this is a fairly extreme case, but I'll give you a bit of context on perinatal stroke in case you're not familiar before we move into commentary on kind of atypical development. So perinatal stroke is a vascular injury, a focal lesion occurring between 20 weeks gestation and 28 days post-birth. It's actually the most focused period of risk for stroke in your lifetime, the perinatal period, which a lot of people don't know. It's the leading cause of hemiparetic cerebral palsy, as lesions are more common in one hemisphere, and they're also more common in the left hemisphere as it happens. You can kind of see that in this heat map of where lesions tend to be across participants. These are different subtypes of perinatal stroke. You can see one subtype covers a large swath of cortical and subcortical brain tissue, whereas in another one's more limited to ventricular space. And perinatal stroke is an interesting model of neuroplasticity. It's an otherwise healthy brain, typically, with a focal injury early in development. So there's a chance to kind of look at reorganization of the brain, or of white matter in particular, compensation in result to an early lesion. So I bring this up partially to talk about tractometry in the lesioned brain. It can be harder to find tracts for, first of all, there could be a lesion that's truncating the tract early. It could be kind of shifting the location of the tract, and that can cause, some of our tools for automated tract extraction to fail or not perform very well. Manual tractography, of course, being the gold standard here, but it can be complex to find tracts, as they may be much smaller, they may be truncated early, they may be shifted by a local or a remote lesion. Even lesions that are removed from a tract of interest can still influence tract structure. I'll touch on that a little bit more in a second. But I did want to mention one tool that I've used quite effectively in perinatal stroke, in case it helps any of you. So there's this RICO bundles tool from the DIPI package that is kind of a semi-automated method to segment tracts. It's based on recognizing streamlines that are of similar shape to the example tracts that you give it, and it performs quite well in brains with lesions like the perinatal stroke brain. So worth mentioning, it's been a pretty helpful tool for me. You can see some tracts that I've segmented on the right here in my own research, typically developing on the top and perinatal stroke subtypes on the bottom. You can immediately see that tracts are present, but they're quite a bit smaller. Like take, for example, this big fluffy arcuate fasciculus in typically developing kids, and the AIS, or the perinatal stroke arcuate fasciculus, is quite a bit smaller, and in fact, in my experience, quite difficult to find. So thanks to RICO bundles for making this process a little more time efficient, of course. Okay, so perinatal stroke I bring up partially to talk about balance in structure between hemispheres, or asymmetry between hemispheres, or lateralization between hemispheres. I think it's a really interesting tool to look at reorganization of the brain. So like I mentioned, lesions are most common in the left hemisphere, and they're frequently limited to a single hemisphere in perinatal stroke. typically asymmetry between hemispheres is minor, meaning that, FA values and MD values of homologous tracts in each hemisphere are pretty similar in typical development, meaning that, there's low asymmetry. But in the case of a lesion in one hemisphere, you would expect, or as you might expect, FA tends to be lower in that hemisphere and MD tends to be higher. That's kind of opposite to the developmental trend we would expect. And that makes sense, right? Say you have an archivesiculus, it's been disrupted by a major lesion. The remaining fibers, there's going to be fewer fibers, most likely. They're going to be, disrupted. Their structure is going to be kind of compromised. That's the word, compromised, by the presence of a lesion. But like I mentioned, even remote tracts that are not close to the lesion can be affected. So we do see lots of differences in asymmetry for perineal stroke participants compared to controls in the archivesiculus. You can see those significance lines all over the place in the left columns here. But we also see differences in the uncinate vesiculus, where lesions don't typically, influence, at least not directly. We still see imbalances in the structure of those tracts. Typically the direction being FA is higher and MD is lower in the contralesional hemisphere, the intact hemisphere, suggesting that, there's kind of a discrepancy in white matter structure between the hemispheres, both for tracts close to the lesion and also elsewhere in the brain. Now, interestingly, to build on this, let's talk about language. So language is typically a left lateralized function. And in this, continuation of the perineal stroke study that I've been showing as an example here, we found relationships between contralesional tract structure and both verbal memory and word production in the arcuate and uncinate. So we're seeing that there's an imbalance, a bias towards higher FA and lower MD values in the contralesional hemisphere, and that the structure of those right hemisphere, typically right hemisphere tracts, are related to language, kind of hinting at reorganization of networks, the language network, to include more right hemisphere fibers to support language function, kind of getting at that, the potential for neuroplasticity in perineal stroke. So this is an example I want to bring up to talk about, differences in tractography, unique kind of reorganization of white matter structure, and also links to behavior. So hopefully that kind of adds a little bit more context to the healthy development trends that I touched on first. Okay, so I also want to take some time in this presentation to talk about, things to be aware of with DTI-based studies. The bulk of our literature is DTI-based in terms of understanding white matter development and links to behavior, and that's good, but it could be better, let's say. So the first thing I want to mention is that, a lot of our research and a lot of the work that I've been talking about is population level, which, means we're taking data from all of our subjects and we're collapsing it to find the big black line here, the population level trend that best represents, healthy development or atypical development, depending on the sample. But this can mask a lot of interesting variability. So if I highlight two subjects from this data set in green and purple here, you can see there's a lot of variability in their FA values for this given tract. It's the inferior frontal occipital fasciculus. A lot of variability in FA between their visits, but also this consistent difference, right? Both green and purple are following the general population trend. Their lines have the same shape as the black one, but the purple subject is consistently a lower FA value for their tract than the green subject. So what does that mean? What's the determinant for whether someone, has a consistently higher FA or lower FA? Is there some link between that, FA discrepancy and behavior or cognitive performance? Maybe, but a lot of the studies that are published out there right now haven't looked at that level of detail. So this is a promising future avenue for research to better understand white matter development and kind of determining factors and features driving development. Looking at kind of inter-individual and even intra-individual variability, I think, is really promising moving forward. And kind of the main point in terms of, complications to DTI-based literature is actual just interpretation of the metrics. So this may not be news to many of you, but for some who might be new, fractional anisotropy and mean diffusivity, or these DTI-based metrics, are sensitive to all kinds of different biological factors. In fact, let me just move on to the next slide to highlight some of those factors. Everything in that red circle, is a biological source of variability, or maybe experimental errors, like a source of unwanted variability. But DTI metrics can be sensitive to all of these features. So if we see FA goes up, what does that mean? It could be a combination of any of these factors changing, along with the fact that, for our typical voxel, we have tens to hundreds of thousands of axons passing through a region. So there's kind of a limitation on how specific our findings can be. But we have other methods available, some new, some old, that can kind of augment our DTI-based findings. So there's many imaging metrics available with varying levels of specificity to each of these individual microstructural features that DTI kind of captures in an umbrella fashion. So just taking myelin as an example, there's every column in this middle figure is a different metric sensitive to myelin, with each bubble showing reports from a different study. You can see there's tons of metrics we could use that are sensitive to myelin. Scatter plot on the right is showing that these metrics share information, but they're not exactly the same, right? Which, I guess I'll kind of pass along in the interest of time here, but getting at each of these metrics is sensitive to myelin in a different way. They may have other microstructural sensitivities mixed in, or different levels of sensitivity and specificity. But each of these measures, regardless of whether they're sensitive to myelin or specific to something else, can be used to kind of clarify DTI-based findings. So I've got two examples here that I'll run through. The first one is a study of white matter development on the top there. So all of these tracks that we looked at had FA increases and MD decreases. But by including measures that were sensitive to axon packing and myelin content, we were able to show that some of those regions with FA increases and MD decreases were driven by increases in axon packing only. Or in the middle, there's a big set of regions that were experiencing increases in axon packing and myelination. So that allowed us to kind of comment on the features that were driving our DTI-based changes. On the bottom, I have another example of findings I reported at a previous OHBM, showing relationships between microstructural features. So there's a myelin sensitive metric in there. There's axon packing or neurite density sensitive metric. There's even a fiber coherence sensitive metric, and each of those things was related to changes in reading related skills over a 2-year period. Now, the really special thing about this study is that we had no FA or MD associations in our sample, and this is longitudinal data. It's change over a 2-year period. So there was no relationship between FA and MD changes in our adolescent sample, but there were relationships with our microstructurally specific metrics. So this really highlights how those microstructurally specific metrics can uncover, trends we might have missed otherwise, or they can help to clarify what's driving our FA and MD relationships that are so well established in previous literature. Now, the burden, one potential problem with having so many metrics to choose from is, how do you kind of leverage these metrics without performing 100 comparisons or 1,000 statistical models and increasing your rate of statistical errors beyond belief? So I want to touch on a technique that can be used to kind of compensate for this, and that's dimensionality reduction or component analysis. So there's this, and before I get started, this is just one of several techniques that could be used to kind of leverage shared information across metrics. But here we are, I'm giving you an example. So there's this great paper that lays out like a really clear framework for how to take many measures with shared information and collapse them into very effective and sensitive components. They use 10 diffusion tensor or diffusion kurtosis imaging metrics and collapse them into 2 components that explained 80% of variance in metrics, in all metrics overall. Now, I use this as a template and expanded to include some non-diffusion-based metrics, and again, successfully found 3 components this time, again explaining about 80% of variance in the sample I was using at the time. So this dimensionality reduction strategy can be used with diffusion-based metrics and other imaging metrics as you see fit. The idea is just to identify metrics that share information and pull out and try to interpret what those components may be sensitive to, what that shared information actually is. Now, both papers that I'm talking about here showed relationships between principal components and age. My study also included an investigation of links to reading behavior or reading performance, which we didn't see any significant relationships there, but that might be due to our sample or other limitations. I do think this is a very promising technique moving forward to kind of leverage the multimodal nature of MRI imaging, and the strengths and weaknesses of various imaging metrics can be combined, or kind of in terms of weaknesses, they can be smoothed over by combination with other metrics. But we already know these components are sensitive to age-related changes. So again, this is a promising method to investigate white matter development moving forward, and probably links to behavior with some future kind of developments. Okay, so with that, I want to leave you with a few takeaway messages. In this talk, I've kind of laid out that we have a foundational understanding established by DTI-based literature of how white matter develops across the brain. I gave you just a little taste of that, along with links to cognition and behavior. We can also use this in atypical populations to investigate kind of the flexibility and plasticity of the brain. Current trends in the field are focused on shoring up and determining contemporary work in the field is focused on ensuring the reliability and reproducibility of reported findings, and also on kind of digging deeper and finding out what microstructural features are underpinning these well-established trends in the DTI literature. And of course, the same levers and tools that we have to improve the reliability and reproducibility of our findings in brain imaging in general also apply here. So using as much data as you can that's appropriate for your project, bringing in multivariate or multimodal imaging metrics and longitudinal data when possible, and also focusing on microstructurally specific metrics to kind of augment the broad measures like FA and MD. With that, I would like to thank the wonderful mentors I've had across my career so far, along with their incredible teams, our funding sources, and of course, the families and children that contributed their time to all of our research. If you have any questions, my email is right there on the screen. And thank you very much for your time.